Eh, And so now it's my great pleasure to introduce Dr. Eran Elinav, who comes from the Weizmann Institute of Science in Israel. And I understand he's traveled far and he's had a pretty crazy, hectic schedule while here in the US as well. So it's a real great honor to have him here with us today. Thank you.
So thank you very much. I'm very happy to be here. I'm going to be speaking to you about the microbiome with association to the sugar and artificial sweetener topics that are discussed here. So my group and I yeah, it is the green one. Let's try this. Okay, we're fine. So my group and I at the Weizmann Institute extensively study the microbiome, mainly the gut microbiome and its interactions with the host. And they say that one picture may be worth 1,000 words, and this is one of these pictures taken from a scanning electron microscopy of a healthy gut. You can see these bumps. These are the villi forming the lining epithelial layer of a healthy gut, and you can see what seems to be like a carpet of shoelaces covering them. This is the microbiome, so nice to meet. I think you can already appreciate how intimate the interaction is between us and our microbiome in the cellular level. And we regard the microbiome and its interactions with the host as a critical component that contributes to metabolic homeostasis or to the development of risk to acquire many multifactorial diseases. When I say we, I refer to my own group and to the group of my good friend and colleague Eran, who is a mathematician also from the Weizmann Institute that performs many of these studies together with us. And we see our mission in life as trying to decipher the molecular language that dictates the crosstalk between us and our microbiome. And I think what's really interesting about it is that this crosstalk is regulated by a number of environmental and host-related factors, such as the examples depicted in this cartoon, which kind of makes the microbiome a hub for all of these signals that can explain their impact on the human host. And given the focus of this meeting, I will be focusing on a few examples of our own studies related to diet and how we try to understand the interactions by which diet regulates the microbiome, and by these regulations may regulate our healthy or non-healthy metabolic homeostasis. So the very naive question that we asked when we started dealing with this is whether we can understand ways of decoding diet microbiome host interactions and their potential effects on the glycemic control, on the normal and hyperglycemic conditions in humans. And when I started reading about what we do now, what's the gold standard, what we do in real life, I started reading all the literature from the early 70s that forms the basis of our current practice and paradigm. And what came out was that almost everything that we do is based on one of several scoring systems which give different numbers to foods, and based on these scoring systems, we and others try to come up with what we call a healthy diet. So for example, calories are one such scoring system which gives numbers to foods, but with respect to the glycemic control, the most widely used scoring system is the glycemic index. So just to remind those of you who don't know what the glycemic index is, it is based on very small-scale studies in the 1970s performed on usually 10 human volunteers that were kindly asked to consume an identical piece of food. So for example, if you would take 10 human volunteers, give them an identical piece of celery, you would then measure their blood sugar levels for 2 hours after they ate the celery. You would probably get a graph like this black graph, and the average number that this graph would represent would be given as the glycemic index of celery. Now if you would kindly ask the same 10 people to eat an identical piece of chocolate cake, their blood sugar levels in average would rise to, let's say, this blue graph, and this higher number would be given as the glycemic index of chocolate cake. Now, if you open up your smartphones, you will find endless tables giving a glycemic index to any food on Earth. And I think you know as good as me that almost any diet on Earth that is aimed at normalizing blood sugar levels is based in one way or another on this glycemic index. Now, here comes the big problem. For some unknown reason, we decided to repeat this experiment, but not to do it on 10 people, but to actually do it on 1,000 people. And when we did this experiment on 1,000 people, and if we focus on the left panel, and we gave the 1,000 people one of several test foods, identical test foods, such as an identical amount of glucose or bread and butter, and we measured their blood sugar levels, we found that the average rise in blood sugar level was exactly the glycemic index of these test foods. However, when we looked at the variability between the 1,000 individuals, it was absolutely huge. The variability was unimaginable. The same was true when we looked at real-life foods. We tested hundreds of different foods, and for each food that we tested, the average response was the glycemic index of the food, but the individualized response varied greatly between individuals. So this already told us that something was wrong in the way that we think. We're kind of asking the wrong question, because if your glycemic index to a given food is opposite than mine, and we want to come up with a diet that would reduce our blood sugar levels, then the same diet cannot be good for both of us. So this concept of a one-size-fits-all diet that we all try to follow is inherently flawed, at least by these results. So this gave rise to a very ambitious study, which we termed the Personalized Nutrition Project, in which we took these 1,000 people and we kindly asked them to give us a week of their life. And during this week, we collected an unprecedented amount of person-specific data, including a very deep and detailed gut microbiome analysis, both by 16S ribosomal DNA and shotgun metagenomic sequencing analysis. We took many blood tests. We made them fill many questionnaires related to food habits, to lifestyle and background medical illnesses, anthropometrics. We developed for this project a smartphone app that enabled each individual to kind of give us in live everything that they were doing during the week of follow-up, when they were going to sleep, when they were waking up, what they were eating, how much they were eating of whatever they were eating. We connected each individual to a continuous glucose monitor, which measures every 5 minutes your blood sugar levels. So from each of the 1,000 individuals, we ended up having over 2,500 accurate glucose measurements during this week. But the real smart thing happened after the week of follow-up, when a very smart group of students at the Weizmann Institute, at our labs, took from each of these individuals all this person-specific data, really an unprecedented amount of data that was collected in this study, and used advanced machine learning techniques which were meant and aimed to accurately predict a person's response to any given food, even foods that were not consumed during this week of follow-up. And to make a very, very, very long story short, this is kind of the gist of the results. And on the left, what you can see is our calculation of the predictability of a glycemic response to food that you get when you go to your dietitian. So there is a sense to the recommendations we get, which are mainly based on giving us a low carbohydrate diet. And we predicted it to be 0.38. But when you use our data-driven, machine learning-based individualized approach, you dramatically increase our ability to predict the glycemic response to foods to 0.68. In machine learning techniques, you have to always use a fresh cohort, freeze the algorithm, and retest your algorithm on this fresh cohort to avoid the overfitting, which we did in another 100 individuals, and you can see that our predictability stayed very stable, which of course made us very happy, but at this stage it was time to put ourselves to the real-life test. So in order to do this, we embarked in a short-term trial in which we took a group of pre-diabetic individuals, and as you know, these individuals already suffer from disturbances in their blood sugar control. They have a 70% chance of developing full-blown type 2 diabetes within the next decade of their lives. And as a clinician, I can testify that we tell them to lose weight, to exercise, they never do, and we really have very little to offer them. So we took this group of individuals, we put them through our process, and then we asked the machine learning algorithm that was, of course, unique for each of them to devise a set of good individualized diets or bad individualized diets for each of these people. We also asked the machine learning algorithm that the good and bad diets for each individual would be isocaloric, so we don't blend the calories. And then we asked each participant to eat his or her good personalized diets for a week, and then their bad personalized diets for another week, while we very extensively monitored them with all the readouts that I've shown you before, including a daily sequencing and measurement of the gut microbiome. And the results were that all of the participants normalized their pre-diabetic blood sugar levels within a single week of good personalized intervention that could, in some cases include ice cream or, chocolate cake. The good and bad components of a person's diet were completely counterintuitive and unpredictable, and were completely unbiased and measured and based on the machine learning. Now, I'm telling it to you in a talk that is focused on the gut microbiome because when we finished this study, we were able to revisit the algorithms and ask the algorithms what they were using in order to get this accurate predictability. And by far, the largest number of features that were helpful in getting a good predictability came from the gut microbiome. In fact, today that we know all of this, we can minimize the amount of data that we collect from a person to the microbiome plus a set of clinical features and get relatively similar accurate predictions. And what we're doing now is we're in midst of a very large prospective, double-blind interventional study in which we take these pre-diabetic individuals, 200, and we either give them the American Heart Association gold standard diet or the personalized approach and follow them for a whole year with all the clinical readouts in order to see whether this new approach would be superior to the one that we're all using and is not very helpful in attenuating or reversing some of the metabolic phenotypes. Now, this brings me now to the artificial sweeteners. So when we were doing this personalized nutrition project, even back in 2013 when we were just collecting the data from this huge cohort, we came across almost accidentally an observation, and this was back when we only had 300 individuals enrolled and quantified, that people who consumed artificial sweeteners seemed to be different as a group as compared to people who were not consuming artificial sweetener in a number of metabolic features, and also they were different in terms of their microbiome composition. And of course, this was a complete naive observation, completely, possibly completely caused by reverse causation. So this by itself does not tell you anything. But for us, it was peculiar and even interesting. And actually, these associations and hits deepened within the years. And or a few months ago, we published a paper based on the same cohort. And in that paper, we aimed to directly compare the contribution of the gut microbiome to the contribution of the host genome to each other and to the downstream metabolic phenotypes in 1,000 humans. In other words, we sequenced the genome of the entire cohort, so now we have a direct comparison of the human genome and of the microbiome in the largest cohort so far, and we are able to directly make this comparison. What we found was that the contribution of the human genome to the variability and composition of the microbiome was very small. It was minimal. We estimated to be 2% of the human variability in gut microbiome is explained directly by the human genome. You can see here an example of a principal component analysis graph of the human genome of our Israeli cohort, which nicely clusters based on ethnicity, Ashkenazi Jews and Sephardi Jews and Arabs. We are a very good country for these kind of analysis. But when you look at the microbiome of these ethnic groups, it's a big mess. There's no clustering. We also performed a million SNP analysis, looking at many different SNPs and carefully correcting them for FDR correction. And we found a very small signal by which the human microbiome is influenced by the human genome. So 98% of the microbiome variability is explained by environmental factors. And when we looked at these environmental factors in our own cohort, there popped our artificial sweeteners. Now, this is a stronger level of evidence because it's a predictor now. It's not just an association as one of the major explanations for human microbiome variability. So together, this gave us some hints that maybe there's something interesting with this particular food substance. Back then, we started looking at a couple of food substances. One of them was artificial sweetener, another was bread. I'm going to focus on the first, in trying to kind of understand a little deeper whether there is a true association, whether there is causality, and whether we can identify some mechanisms explaining it. So we started with